Let's see, is this microphone working? Can people hear me? Okay, perfect. Yeah, so I mean, we're really interested both in continuing to develop methods that push the limits of what we can do with Markov models, and also, doing a lot to actually use them to guide experiments to understand really challenging systems to prove the utility of these things and help figure out where we need to improve the methods also. So we work on a range of systems, but one of my favorites is a class of enzymes called Betalactamases. And these are really important players in antibiotic resistance. So bacteria will express these enzymes, and they're really good at binding to and degrading beta-lactam antibiotics like penicillin at their active site here. Right, so there's a lot of interest in learning how these enzymes work and developing ways to inhibit them so that we can, say, restore the efficacy of existing antibiotics, because expression of these enzymes turns out to be the most common form of antibiotic resistance. So they're also really interesting to me because we have a great interest in understanding the role of proteins' excited states and functions, and new opportunities they open for understanding processes like allostery and guiding the development of allosteric drugs. And the beta-lactamases are kind of an interesting choice for someone interested in those sorts of questions, because the conventional wisdom is that these aren't allosteric systems, that these are systems where you can really think about the protein as largely adopting this native conformation we see via crystallography. But there's some tantalizing evidence that there's more to the story. So in one study that I really love, people were trying to computationally design new beta-lactamase inhibitors. So they performed large docking screens and found some interesting looking compounds that they then showed actually inhibited the enzyme experimentally. But when they went to solve the co-crystal structure, they got a bit of a surprise because they got this yellow structure with the compound bound here. Right? So the compound didn't bind where they expected it to in the active site. Secondly, where it binds is a cryptic pocket in the sense that there's no pocket here in the apo structure in blue. And thirdly, not much has really changed about the active site, so it's not clear why binding of this compound should inhibit the enzyme in the first place. So when I saw this coming from a background in protein folding, I thought, man, I'll bet this sort of thing is happening all over the place, and that if we had methods for essentially sitting and watching the sorts of dynamics that beta-lactamase undergoes at thermal equilibrium, we would see things like these helices spontaneously pop apart, and maybe other motions that we could then go and intentionally try to target instead of being surprised by them on occasion. So we've been doing a lot to develop Markov state model methods for using molecular dynamic simulations and MSMs to do this sort of thing. I apologize if this showed up, but the basic idea is that we're going to represent the underlying energy landscape that controls a protein's thermodynamics and kinetics with this network model, where we essentially, you can think of having states that roughly correspond to minima in this high-dimensional energy surface. And what we want to do is use our molecular dynamics simulations to learn both the identities of these states and the rates of hopping between adjacent states. And this is extremely powerful because now, even if we have simulations that have a limited length compared to the sorts of timescales we'd like to access, as long as we can start them all over the landscape and independently learn these different transition rates, now we can piece all of these relatively short simulations together into models that capture long timescale events long beyond the reach of any individual simulation. So just to show an example, we've done a lot of work with showing that with these sorts of things, we can watch beta-lactamase's spontaneous fluctuations and see things like the cooperative formation of this cryptic pocket, such that hypothetically we could have realized it was there and gone and targeted it intentionally. And we can also start to experimentally test our predictions, right? So we've also seen other cooperative opening events, which then raises the question of, are these viable drug targets or not? And going all the way from simulation straight to drug is a tall order, so we'd love to be able to verify aspects of our predictions without going all the way immediately to small molecules and having all of the different caveats that come with different steps towards drug discovery. So one of the things that we've developed is this thiol labeling assay, where if I play this movie again, you'll see that there's this cooperative opening. So there's these residues that are buried in the apo structure but get exposed to solvent by the opening of this pocket. So what we'll do is take one of those residues and express a new variant of the protein that has a cysteine at that position, which is one of the most highly reactive amino acids. And now we can introduce a labeling reagent that will form a covalent tether with that cysteine if it gets exposed to solvent. So here we're doing that for one of the residues buried in this pocket. You can see that we get this nice single exponential labeling consistent with this cooperative opening motion. Here's just the residuals showing the single exponential as a good fit. So that tells us that this thing is getting labeled, so that's encouraging and supports the idea that there's this pocket. But you could say, well, maybe it's due to something like unfolding, right? So we also have to do a bunch more experiments and controls. So for example, we'll do our labeling experiments at a range of concentrations of our labeling reagent. And now we can analyze this with the same machinery that's used for analyzing hydrogen deuterium exchange data, if you're familiar with that. But basically, in this case, our observed rate of labeling has this linear dependence on our concentration of our labeling reagent, which tells us that the slope of this line is giving us information about the equilibrium population of the open form of this pocket. And so a relevant control is to make sure that that population isn't equivalent to the unfolded population at equilibrium. So we've gone and done the equilibrium unfolding experiments, and this dashed line down here is what labeling we would expect if labeling was due to unfolding. So we can see that the observed labeling is way faster than we would expect from unfolding, which helps to support that this is due to a fluctuation in the native state and not some other process. Okay, so we've experimentally verified a couple of pockets that we found in beta-lactamase, and now the question is, okay, can we actually use this for drug design? And one of the challenges here is that we've got lots of structures, right? Like our model for beta-lactamase, this particular beta-lactamase has something like 5,000 states. And so I didn't feel really comfortable cherry-picking one state and saying, okay, we're going to plug this into our favorite docking tool and go from there. So we've also been developing twists on docking, one of which we call Boltzmann Document. So the idea here is that we're going to take a representative structure from each of these states in our Markov state model. We're going to dock our library of compounds against each of these states to get a docking score per compound per state. And now we also know the equilibrium populations of these states. So we can take a dot product between those populations and our docking score for each compound to get a Boltzmann-weighted docking score. And this is really nice because it can balance the fact that we'd really like to see high affinities between our compounds and structures, but that may not do us all that much good if the protein structure is really, really high energy to start with. And so this provides a natural way of balancing between trying to find things that bind tightly to one structure or bind moderately tightly to multiple structures, and what the energetic cost of stabilizing a particular conformational state is. And we've gotten some promising results with this. So here's an example of one structure where we've taken some of the states from our Markov model where this pocket that I've showed you before has opened. So here we're looking down on the active site, and here's those two helices that separated before. I changed the view because I wanted to highlight that in some ways you could think of this as the same pocket as I showed you from that crystal structure at the beginning, but I would argue in many ways it's really a different pocket because this compound that we've modeled in here is reaching back into this huge extension of this pocket, back behind these alpha helices here and these beta sheets here, right? So there's a lot of different interactions that this compound is forming that we still wouldn't have guessed were there from the crystal structure that I showed you before. So we've done the follow-up experiments and can show that compounds like this actually modulate the enzyme's activity. When we started this, we presumed that, well, someday maybe we'll understand beta-lactamase's mechanism well enough that we could design activators if we wanted to. But in the meantime, if we're just finding things that bind one of these cryptic pockets, it's much easier to break things than to make them work well. So we're probably going to find inhibitors. Much to our surprise, so far we've found 3 compounds from our computational screens that have stood up to our experimental tests. And 2 of them turned out to be activators and 1 is an inhibitor. So activating this enzyme was actually a little bit easier than expected it to be, which has interesting implications for future work, right? Because we could start to think about doing things like using allosteric modulators to reverse the effects of deleterious mutations, for example. And in addition to finding these pockets in conventional targets like beta-lactamase, maybe we could find these sorts of pockets in proteins that are currently considered undruggable. Right, so this opens up all kinds of potential applications. In the spirit of a meeting where I think people do a really good job of talking about both the strengths and limitations of their work, I thought I'd acknowledge these aren't really the greatest lead compounds necessarily for a drug discovery program, right? So here's our compounds and our dose response curves, and you can see that the half maximal effective concentrations are in the tens to hundreds of micromolar, right? And this is interesting because it means these compounds might bind pretty tightly actually to some of the states that we're targeting because they're pretty high energy, so we pay a penalty for stabilizing them. But certainly if we want to come up with anything that anyone would consider using as a lead compound for drug development in the future, we'd like to find more potent things. So this has inspired more methods development that one of my students, Justin Porter, will talk about later on that has some promising preliminary results. But nonetheless, this was super encouraging that we can find these pockets in proteins like Betalactamase and experimentally verify that a pocket exists and has the capacity to exert allosteric control over activity, and we can even do this with small molecules. And we've done a number of things like showing that mutations to critical residues that these compounds interact with. Now the compound doesn't work anymore, and mutations elsewhere don't have an effect. So this is looking pretty good. And has emboldened us to start looking at some new targets. So it's interesting to talk a little bit about looking at proteins involved in Ebola infection now, because this is scientifically interesting because Ebola only has one enzyme in its genome, and the rest of the other 6 proteins in the viral genome predominantly interact with each other and host proteins through protein-protein interactions, which are really hard to drug. And as I was coming in this morning, actually, on the news, they were talking about a new Ebola case in a large city in the Congo. So it seemed timely to talk about trying to treat these sorts of infections. So this is really interesting also because, like I said, there's only 7 proteins in Ebola's genome. And so in order for the virus to function and survive, they're all really highly multifunctional. So this one domain is a domain from a larger protein that has a couple domains separated by flexible linkers. And just this one domain plays an essential role in the viral capsid. It's required for transcription of the viral genome, and it inhibits the host innate immune response in at least one, but probably multiple ways. Right, and it's only about 120 residues. So the more and more interactions you tack on to this thing, the harder and harder it is to imagine that there's really all these different distinct binding interfaces that perform these different functions. And so we were curious to explore whether this protein might exist in an equilibrium between different conformations that are primed for these different functions, and that if we could get in there and see those different conformations, we might see things like cryptic pockets and have an opportunity to manipulate this equilibrium in a way that would inhibit viral replication or proliferation, for example. And this has been pretty encouraging. So here's another morph just showing this large motion of this loop down here that creates this cryptic pocket here that was tantalizing to us because we think that this protein interacts with another viral protein at this interface here, and we know that it binds RNA to protect the ends of double-stranded RNA over here. And as kind of an aside, in a paper, my collaborator had mentioned that there's this negative cooperativity between these interactions. And so we think that this motion might explain how this communication occurs between these two sites. We think maybe the open form is able to bind this other viral protein, and the closed form binds RNA. And that if we could get in and find small molecules that bind here, for example, we might be able to shift this equilibrium and, again, as I mentioned before, inhibit viral replication. So this is still in a project that's in the early stages, but as a first step, we've started applying our thiol labeling technique. And this is a convenient case because it turns out there's 4 native cysteines in this protein. One of them seems to remain buried throughout our simulations. One is surface exposed, even in the crystal structure. But these 2 guys down here are buried in the apocrystal structure and get exposed to solvent by the opening of this cryptic pocket. So we run our thiol labeling experiments, and we see labeling of 3 cysteines, which we're reasonably confident are this guy up here and these 2 down here. So that's encouraging and is leading us to do more and more follow-up experiments. But as with the beta-lactamase stuff, these initial successes have also spurred us to keep pushing the limits of what we can do. So we've also been tackling some even more challenging systems. So one that we've been spending a lot of time on recently is a class of proteins called G proteins, which you might recognize because they're the namesake of the now more famous G-protein coupled receptors, or GPCRs. So these are molecular switches that play really important roles in a variety of signaling cascades involved in everything from vision to olfaction and neurotransmission. And the way they work is that you can think of the default state as being this GDP-bound inactive state, and a GPCR will come along and catalyze an exchange of GDP for GTP, and that will transition this protein to its active state, and that will dissociate from GPCR and go on to interact with downstream effectors and trigger intracellular responses. And this was interesting to us for a number of reasons. One is that if you look at the GDP and GTP bound conformations of those proteins, they're very similar, and both basically completely encapsulate the nucleotide. Right? So somehow this protein has to open up for nucleotide exchange to occur, and then readopt a very similar conformation once GTP has bound. But the GPCR doesn't work in a really obvious way. Right, so one might think, like, oh, maybe it might bind at this hinge, and there's a really simple explanation for how it catalyzes this exchange. As it turns out, the GPCR binds at the top of the protein from this orientation, and this allosteric signal has to propagate over 30 angstroms to impact the nucleotide binding site and catalyze this exchange process. And it's been really difficult to understand how this works from a purely experimental perspective. And it's also a huge challenge computationally, because this protein is something like 300 residues, and even the GPCR catalyzed exchange is hundreds of milliseconds. Just to give you a little bit more information before I dive into what we've been doing and what we've been learning, there's a lot of structural elements that are nice to be able to talk about for this thing. So there's a convenient naming scheme where like H5 refers to helix 5, so that's this long helix in the back that's vertical. S1 refers to beta strand 1. And then two other elements I'll bring up are the phosphate binding loop, the P loop, and H1 contacting the nucleotide here. Okay, so there's been lots of interest in understanding how this conformational change happens and how it catalyzes nucleotide exchange. People think there's multiple intermediates that are involved, and we've seen one of them by crystallography. Right, so in this structure, the green structure now, you can see that it's not shown up, but GPCR binds up here, and it facilitates this translation of this large helix 5 in the back here, and you can see that this helical domain down here is swung way out to expose the nucleotide binding site. Right, so it's tantalizing, and people have speculated like, oh, there's this translation of H5, and that causes some disorder in H1 down here and helps favor opening of this helical domain. But then there's a lot of contrary experiments and simulations that say, well, actually these domains are fluctuating open even in the absence of GPCR, and there's all kinds of questions with any crystal structure, especially with ones with as many modifications as this one. So, and we think there's multiple intermediates, not just this one. So what else is going on, and how does this process work? So to get at this, we've been combining a couple of different methodologies, like metadynamics and Markov state models. So the idea is that we're going to use metadynamics to get some initial potential pathways for GDP release. So that would work, say, if you start simulations down here and you want to see, exchange and going over to this state. With metadynamics, we'll run some simulations and keep adding these Gaussian potentials to your free energy surface that push you away from states you've seen so far. So anyway, starting simulations here, adding some Gaussian potentials, and they'll push you out, help to encourage you to explore other regions of the free energy surface. So this is really valuable for seeing rare events, but there's some challenges with then extracting kinetic and mechanistic information because you're changing your free energy surface. And so one of the nice advantages about Markov state models is that you can get both the thermodynamics and kinetics for your system and really delve into these mechanistic questions. So what we'll do is use metadynamics to get some initial pathways, and then take structures along these pathways and use them as starting points for conventional molecular dynamics simulations that will then use to construct a Markov model and watch how this process of allosteric activation and GDP release unfolds. So we've done this, and we can do a number of consistency checks with experiments. So here I'm just showing the distribution of distances between two residues lining this the ends of these two domains here to look at domain opening. So you can see that consistent with some previous simulations and deer experiments, there is significant opening occurring even in the absence of, or even when the GDP is fully bound. And as we progress through the release process and look at the rate limiting step, we do start to favor more open structures. And once we've released, the protein is continuing to exchange between open and closed structures. So these sorts of consistency checks are encouraging that the model that we get at the end is a reasonable reflection of reality. We can also now go into our Markov state models and start using things like transition state theory to look at or transition path theory to look at the highest flux pathways for GDP release and start trying to learn what the key steps along the way, and particularly the rate limiting step for this process is. And that's been really interesting because we can look at both changes in conformational disorder and structural changes by pulling out these states, rate limiting steps along these highest flux pathways. So here I'm color coding the structure by changes in conformational entropy. So red is increase, blue is decrease. And we see some things that people might have expected based on what they reported in the literature, like this increase in disorder in this helix 1 or H1 that people have postulated based on B factors and missing density and the one available crystal structure. But there's a lot of other interesting changes, and in fact the whole nucleotide binding site in general seems to have had an increase in conformational disorder that people really haven't talked about that much. And if we start looking at the structures, this also reveals some interesting differences between the general conventional wisdom in the literature. Right, so here I'm showing an overlay between the GDP bound state in blue and what we think is the rate limiting step for GDP release in orange. And you can see a couple interesting features. One is that the domains have only opened a little bit, right? So it doesn't take that much room. The domains don't have to swing all the way out for nucleotide to get in and out of this binding site. And instead of seeing translation of this H5, helix 5, this long vertical guy in the back, we've actually seen it tilting, which is definitely a strong contrast with this standing universal mechanism for G protein activation that people have proposed, which really centers on translation of this helix. And so what we think is happening maybe is that this is actually an earlier step than what's been seen in the crystal structures GDP can release, and that maybe this state that we've seen crystallographically is a subsequent more stable intermediate that might actually play a dominant role in facilitating GTP binding rather than promoting GDP release. We can also get in and start making sense of how this signal is propagated. So, as I showed you, these changes in entropy are really important. So that's hard to get at with some of the standard tools for inferring allosteric communication from simulations. Because these really focus on things like concerted structural changes between different rotamers in the protein. Imagine if the blue dihedral changes rotameric states, the red rotamer is also highly likely to change rotameric states. But this might not be really good at capturing these things like some conformational change happening up here and inducing disorder in this binding site. So we've also borrowed some really interesting mathematical machinery from David Chandler's work on dynamic heterogeneity to start classifying dihedral angles as being in ordered and disordered states and looking at correlations between both structure and disorder. So here in this case, a hypothetical example where you can see when one dihedral is rapidly hopping between different rotameric states, another dihedral is essentially locked into one rotamer and vice versa. So we can find the whole spectrum of correlations between structure and dynamical state of one dihedral and structural and dynamical state of another dihedral. The math is fun, so I'd encourage you to go check it out, and I'm happy to talk about it later. But the end result is that we can take all of our simulation data and we can start asking, who's correlated to these elements, structural elements up here that we know interact with GPCR, and then once we find those structural elements, ask who they're correlated to, and work our way to the nucleotide binding site to understand how this signal is propagated. And the network that we found is really interesting because there's definitely a strong component that's due to the aspects people have focused on, like this helix 5 communicating with helix 1 and the guys in pink here that directly interact with nucleotide. But there's also a really important role for some of the beta sheets, for example. And this maybe helps to resolve a standing conundrum in the field, which is that the things that we see move crystallographically are things like H5 and H1, but there's a lot of evidence from other experiments like NMR that see chemical shifts in these beta sheets. So by being able to capture both these concerted structural changes and changes in disorder, we can start to piece all of this together into a model that takes into account and is consistent with a wide variety of experiments. And it looks pretty promising. So there's some interesting things. Like I said, we see tilting of this H5 to from this blue structure to this orange structure. The green structure is the translation that's seen in the crystal structure that I showed you. It turns out there's another crystal structure with a fragment of H5 bound to a GPCR, and that's both translated but also tilted to the exact same extent as our structure. So that's kind of tantalizing that both of these things may have a role to play. And we can start to get into details that have been hard to probe experimentally. So one of the things that we think promotes disorder in this helix 1 that is so important is the breaking of this pi stacking interaction. And people had speculated that might be important based on structure gazing, but it turned out to be hard to probe experimentally because one can't just zoom in and watch these things happening. And in this case, mutating these residues has such a profoundly deleterious effect on protein stability that you can't work with the resulting proteins, but we can get in and see what they do. Okay, so hopefully giving you a sense that we're making good progress with finding and drugging these, or targeting at least, these cryptic pockets and starting to see them in a variety of different proteins that one might be interested in allosterically modulating, and starting to get into the mechanisms of these really challenging conformational changes that would have been hard to imagine tackling years ago without all of this technology. With that, I'll briefly thank my group, and I'll be happy to take questions.
Yes, Dolly.
Yeah, I wanted to ask a question about your Boltzmann docking for Betalactamase. Or the modulators that you found, did they bind to many low populated states, or was there just one dominant state that if you had identified it correctly, you could have just got to that state?
Yeah, so a couple of states contributed, but they were pretty similar, so it's hard to give a definitive answer to that, I guess. I think multiple states contributed, though. But largely the structures they bound to, at least of that pocket, were very similar, and lots of the changes were outside of that pocket.
Questions? Dan, then New.
So, very nice talk, and your approach is like a lot of modern, I would say, pipelines use a few different techniques, and I'm just trying to keep my eye on the ball and figure out where the main advantage is coming. So, for example, in the cryptic pocket finding, so I guess you were analyzing a set of molecular dynamics trajectories that are exhibiting these pockets, right? So, did you ever just kind of quantify the extent to which the MSM per se is helping you find the important structures, or if you had just instead put it through a pocket finding filter and use that to dock?
Yeah, so the MSM is particularly useful for finding those structures, and then for sampling the structures. So Max Zimmerman from my lab will talk about some of the sampling methods that I didn't get into, but that help accelerate the discovery of these structural states compared to just using long molecular dynamics simulations. So the MSM is really helping discover the structures, and then once we have the MSM, we're doing things like applying different pocket detection algorithms to figure out which states of the MSM are interesting. And Justin will talk about some of the methods for then analyzing the data. And so, because it turns out, so the MSMs are really useful for seeing these slow conformational changes, but once we hypothetically capture them, we have 10 million protein structures or whatever sitting on a hard drive. So
But is it
it's
the
still a
MSM?
challenge to pull that out. And the MSM doesn't immediately answer that question. So it's secondary layers of analysis of the MSM to tell you where the pockets are, for example, what they look like.
MSM find, you're using a strategy to spread out trajectories in space. Is that MSM per se? I guess that's sort of.
Yeah, so some of the initial work, we actually just ran like 1,000 independent simulations, started from the crystal structure and let them go. So the MSM really wasn't, in the initial things, adding a lot to the sampling, just helping us analyze the data. But now we're doing these adaptive things where we run some simulations, build the MSM, and then spawn new simulations from interesting structures. And like I said, Max will get into more of the details on that, but now we're using the MSMs in a more prospective way to drive the sampling.
Maybe then Chris?
I missed something
Mhm.
with the Boltzmann Document, but
Mhm.
let's say you have one drug and it binds really well to one state and really poorly to another state.
Mhm.
Its score, it will average out and you won't be able to find it.
Mhm.
Does it not scare you that there's like a large distribution of drugs that would be found by otherwise just doing like this massive matrix calculation instead of averaging out that would be more useful in these things?
Right, so I mean, other people have done what they call ensemble docking, where they just dock against lots of structures and take the best score against any of them, right? So that would be, in my mind, the obvious alternative approach, but then that scares me more because it doesn't account at all for what the cost of stabilizing the state that you get a high score against is. So we're trying at least to account for that in a systematic way. But yeah, I agree. It's challenging, but I mean, yeah.
One more quick question from Chris.
Yeah, so I suppose I have millions of compounds and I want to do this on ensemble document, or the Boltzmann document approach. The question is, have you found any useful approaches to enrich your protein conformations in the MSM for a subset that are more likely to yield these productive protein ligand interactions, such as druggability scores or something like that? How do you pick the ones to use? Because it's more economical, let's say, dock 20 or so conformers.
Right. So maybe this also touches on Mandeep's question, but in practice, what we'll do is apply pocket detection algorithm to all of our states, pull out the subset of states where some pocket is opening, and dock against those. So we really focus things, and the 4,000 states that the pocket isn't open don't contribute to the average we actually calculate. So that helps us focus in on things. And yeah, we are starting to use different metrics for druggability of pockets to prioritize things.
All right, let's thank Greg again.